Dr. Colternell. Thanks, organizers. Okay, I'm going to talk about my postdoctoral work in the group of Johannes Söding. So we developed a tool that aims to reconstruct lineage tree topologies using single-cells RNA-seq data. So as I think you are all already convinced that single-cell transcriptomics is a revolution in our capabilities of understanding molecular biology, because we can just having a developmental process like this one in which we have time involved and from which we can observe the emergence of different cell types from a population of progenitors. We can just sample at different stages in the process, and we can just measure for each cell how many copies of transcripts we will have for each gene. So we have at the end one matrix, which is what I get, a gene expression matrix in which we have detailed all this information. So in order to make some sense of this, we need to transform the data and try to embed it in a new space that, given some coordinates, there are many ways of doing this. And we can have this distribution of cells, and we can make some sense of them in a hierarchical way. And once we have this tree, we can just classify the cells, given some markers, and make sense of them, and try to understand which are the genetic differences among them. So this can be tackled in modules. The first one is we need to transform the data and then reduce the number of dimensions. And then we can reconstruct the tree that represents the cells in this new space. And then we can do a lot of downstream analysis, like pseudotime assignment. We can try to see which are the differentially expressed genes in different cell types and try to reconstruct gene regulatory networks and so on. So for the dimensionality reduction, we don't really work on this. There are amazing tools for doing this. So there are different techniques to do this transformation of the data from the gene expression space to the manifolds and then take the first dimensions that are the most important. So we are going to just use software that is around there. So for example, Monocle 2, Destiny, or Slicer. We are going to transform the data and take these coordinates, take the most important ones, and then just to give them to our tool to perform the lineage tree reconstruction. So I'm going to show how do we perform with this in this case study, in which these authors studied how zygotes develop into blastocysts. So they take the embryonic development and they take samples at different stages up to 64 cells. And this is a qPCR data set, so the number of genes are just a few, but they are very, very accurate and very confident about the values we get. So we have this matrix with this number of cells and this number of genes, and they will just calculate the diffusion map from this matrix. So the diffusion map, if you take the first 3 dimensions, looks like this. And we are going to represent this in 2 dimensions just to improve visualization. So it's just transforming the third coordinate to the plane. So we have something like this. So our objective is to try to reconstruct a lineage tree topology from this, given an embedding and a reduction of dimensionality. So in order to do that, we apply some steps. The first one is try to construct a scaffold tree. So given the embedding, we are trying to first check where are the endpoints, where are the cells that represent the different cell types here. So we calculate the Dijkstra with the shortest path distance in between all pairs of cells, and then we can just take the one that maximizes this measure. So this is going to be our first pair of endpoints. And after that, in order to check which are the other endpoints, we're going to calculate for all the cells that are not in the path in between these two, the one that maximizes this measure here, which basically means the cell that maximizes the length of the new branch that we are trying to include in the pre-existing tree. So we apply this until we cannot include any more cells, such that the new branches are going to be longer than the square root of n, with n being the number of cells, or until we fulfill the number of cell types we want to detect in the data. So once we have the endpoints, we need to find where the branch points are. And in order to do this, we apply the neighbor joining algorithm in order to do this. So, oh, sorry, this is cut here, but this is the neighbor joining you can find in Wikipedia. I think you can just find it quickly. So we are going to detect which is the pair of endpoints in the data set that are the closest ones. So it's the pair that we are going to try to join through a branching point. So then we minimize this function here, which is just trying to see which is the cell that better explains the bifurcation between these 2 endpoints. And we can do this iteratively until we can reconstruct just the full topology. So now we know which are the endpoints, which are the branch points, and also super important, how they are connected. So if we apply this on our data, we have this here. This is very robust in all the cases we have tried. but this is sparse. I mean, the density along the tree is not homogeneous, and there is a poor interpolation. So in order to do this, we are going to apply principal elastic trees, which is similar to what Trapnell presented in his monocle paper. It's just a principal curve something algorithm. So we are going to initialize this algorithm with our scaffold tree. That's what we call it, scaffold, and we just have something like this. Now we are introducing 100 nodes to interpolate the data here, and then we can assign all the cells to the different tree segments, and given markers in the matrix, we can just assign the different cell types. So now we want to study gene expression along the reconstructed tree. That's why we did this. So what we would like to do is, if we have some gene, we want to study how this gene is expressed in different parts of the tree. So we would like to see something like that. Given a pseudotime measure, which likely I don't have to introduce, we want to see how the expression is in the different cell types that we detected in the tree. So in order to do that, we just calculate the pseudotime as Trapnell does. We define a point 0 in time, and then all the others take a pseudotime value which is proportional to the distance to this point. And then if we just make a picture, a graph of the expression of the genes in the matrix according to pseudotime, we have something like this. Of course, you can see some patterns. These colors are corresponding to different cell types we detected, but sometimes it's very difficult to see. So what can we do? So we can just take the structure we detected, and under the assumption that this lineage tree structure is maintained in the full dimensional space, we're going to try to embed this tree in the gene expression space in order to see how to interpolate all the dimensions in the full space. So we are going to apply elastic trees again, giving as an initialization the tree we just detected. And first, we need to start from somewhere, because the nodes in the tree are fake cells, so we need to give them some initialization point. So this initialization point is going to be just the average expression value from all the cells that are close to each of the nodes in the tree that we reconstructed. And for those nodes that are in empty regions in the space, we are going just to give a null vector, but we know how they are connected in the space. So this is also very important. So once we apply the elastic trees algorithm, we can smooth to something like this. So now if we again make these graphics of gene expression as a function of pseudotime, we have something like this. So now we can make sense of how the different cell types express the different genes given the initial matrix we have. So there are different methods, of course, to do this. One is monocle 2, the other is slicer, and there are others, but we consider these are the best ones. So, but the embeddings, they look different. So it's very difficult to check, or at least for me as a non-biologist, to see which is the right embedding. So in order to do that, Nico, he just produced this software, Prost, to simulate gene expression matrices, and we can tell the algorithm which is the topology we want to recover after we produce an embedding. For example, these are examples for simple, double, and triple bifurcations, and we produced sets of 100 of simulations with up to 12 bifurcations, and for all of them, we produce the embeddings using slicer, monocle, and malloc. So when we do the benchmarking for the branch assignment of the cells, because we know from the input to which branch each cell belongs, this is what we get. So in the beginning, in the simpler topologies, there's not that clear which is the best tool to doing that. But for really complex topologies, then we are making some difference. Unfortunately, the benchmark for the pseudotime, we don't have it because we would love to discuss it with you, because it's not really easy to benchmark pseudotime in complex topologies when you have a lot of branches and bifurcations. But we are trying to work on it. So yeah, that's it. So concluding remarks, we have a very robust tool to reconstruct lineage trees in very complex topologies with up to 12 bifurcations. That's what we checked. We can reconstruct the expression profiles as a function of pseudotime by interpolation of the full expression space. And given a tree, it's very easy. I didn't talk about it, but this is the Pulse dataset for blood developmental. We can just, given the segments, say, okay, give me a test, a statistical test, and give me which are the genes that are important in the different branches in the tree. This is very easy once you have this structure. So I want to acknowledge my collaborators, Nico, who is around here. I think he's recording the session. Jacob, who is a rotation student that worked with me. Laura was a master student, of course, my advisor, Johannes Söding, our collaborator, Barbara Trollten, and the entire group, and of course, my funding agency for my fellowship. And thank all of you for listening.
So the question is more to these methods in general. In how far does one account for the sparsity of the RNA-seq data in a single cells, and how much difference does it make on the results?
Well, first of all, we are not working in the problem of selecting which genes we are going to use, because this is very well done. But, I mean, we are not working that part. We are taking for our results very high quality and data sets that we know other people used. So we are trying to avoid this because we want to test our tool in cases that are kind of easy to solve. So yeah, we are not working that part. But, yeah.
More questions? Maybe I can ask a next question. I was wondering, did you investigate what type of genes would be more often regarded as markers to find branch points? Is there a particular bias towards particular like transcription factors, signaling molecules, or highly or lowly abundant genes, which come more often as markers to find the actual branch points?
Well, we haven't investigated that much. What we observe, for example, something super interesting, that cell marker genes, for example, they are expressed in very low copies, and they are usually the ones that pick up in the branching regions. If you analyze some data in which you have a progenitor population of cells. But no, I mean, right now we are trying to go a little bit into that, especially because sometimes it's not easy to define a cell type by one or two genes when you need a network, a non-redundant network, and this is a combinatorial problem, so we are not still there. Yeah.
So I'll ask you a version of the question that I got. How do you, absent some kind of a principled likelihood model for a tree, which I think you don't have, we certainly don't have, how do you select hyperparameters for the algorithm?
Well, for the scaffold part, we don't have too many parameters. The only parameter we have is the threshold that you say, okay, how long the branches need to be in order to be valid branches. And then, yeah, it's just that you can also say, okay, I want 5 branches. But then for the elastic tree, this is a bit tricky because it's an energy function, and then also minimizes the error in between the nodes in the space and the cells. And this has 2 parameters basically, mu and lambda, which are 2 parameters in the terms of the equation. We just fitted these values given real data sets to which we were able to reconstruct meaningful trees with up to 200 nodes in real data sets. But of course, sometimes it can fail, and then you need to tweak these values and of course the number of nodes. Yeah.
Thanks.
Let's thank the speaker again.